Hello everyone, this is Mr. Sachin Kharade, Assistant Professor, Kekewa College of Agricultural Biotechnology, Nasik. Now going to deliver a topic on microscope. So the basic definition of microscope is microscope are instrument designed to produce magnified visual or photographic image of small object. So the microscope must accomplish three tasks. First one, produce a magnified image of the specimen. Second, separate the details in the image. And third one is the render the details visible to the human eye or camera. So these three basic tasks which is accomplished by this microscope. So we will turn towards history of microscope. So the scientist Hans and Zacharias Janssen, which is Dutch eyeglass maker, or who invent first compound microscope which having two lenses and tube with lenses at each end. Second one is the Robert Hooke, who is the English chemist, mathematician, physicist, and the inventor of compound microscope. He he do the some improvement inside the compound microscope. Third scientist is the Anton von Leeuwenhoek, who is the wine assayer, surveyor, cloth merchant, minor public official, and the inventor of simple microscope. So he invent this simple microscope which having one lens. Then the basic principle of an microscope is, so this is the classical example of compound microscope which having the focal plane and image plane. So the light which is passed from the objective and which is passed from the image plane towards our eyes. So we can see the specimen or the object by our naked eyes. So these are the structure of an microscope which having the basic parts such as body tube, nose piece, objective lenses, then stage clips, diaphragm, then light source, then the ocular lenses, we can see the eyepieces, then the arm, then stage, coarse adjustment, fine adjustment, and the base. So these are the basic parts of an microscope which having the different functions of each part. So there are three types of microscope generally used by the researcher. First one is the simple microscope which having the only one lens. Then the second one is the compound microscope which has two set of lenses. It can be magnify things that is 100 to 200 times larger than they really are. Then third one is the electron microscope can magnify the objects up to 3 lakh times or 3 lakh x times. They do not use lenses but use electrons to enlarge the image. So we can see the parts of our microscope one by one. First one is the ocular lens. We can see, or we can call it as a eyepiece, the lens of the microscope that you look through, we can call it as a eyepiece. We can see. Then the second one is the coarse adjustment, the large knob on the microscope that you turn to bring the object into the focus. We can call this knob called as the coarse adjustment. Then the fine adjustment, you can see the small knob on the microscope that brings the image into the focus. We can call it as a fine adjustment. Then the basic part, that is arm, which is the support or the part of microscope which is used for the supporting the body tube. Then fifth one part is the body tube, the part that hold the eyepieces and the objective lenses. Then sixth one is the nose piece, the part at the bottom of the body tube that hold the objective lenses and allow them to be turned. The seventh part of microscope is the high power lens or high power objective lens, the lens that magnifies the object the greatest amount, usually 40x. Then eighth part is the low power, that is scanner objective lens, the lens that magnifies the object the least amount, usually used to find the object magnifies only 3x or 4x. Then the ninth part, that is middle power objective lens, the lens that usually magnifies the object more than the scanner lens but less than the high power lens, usually 10x to the 20x. Then the 10th part, that is basic part, that stage, the flat part below the objective lenses where the slide is placed to see. Then the 11th is clip, the part that hold the slide in place so it doesn't move here and there. Then the 12th part is the diaphragm, the part that control the amount of light entering the field of view. Then the 13th part, that is light source, which is most important part of the microscope, the lamp or the mirror under the stage that send light through the object being viewed by our naked eyes. And the 14th part is the base, the bottom part that supports the rest of the microscope. We can call it as a base part. We have ocular lenses, then the body tube, then objective lenses, stage clip, then diaphragm, then the light source, then the arm, we can hold the microscope, then the stage, which is used to put our slide, then the coarse adjustment, then the fine adjustment, and the base, which is very much important for the microscope. So here is one important note is always carry the microscope with one hand holding the arm and one hand under the base, which is most important. Then the next one is the field of view. What is the field of view? Field of view is the area, that is circle, that you see when looking through the eyepiece. Then the comparing the power of magnification, we can see better detail with higher the power of magnification, but we cannot see as much of the image. So in both image, which of the which of this image would be viewed at the higher power of magnification? This is the question. Then next one is the what is power? To calculate the power of magnification, multiply the power of ocular lens by the power of eyepieces. So ocular lens power is the 10x and the objective lens power is the 40. So with this introduction of microscope, we surely know what is microscope, then what is the part of microscope, what are the types of microscope, and the what is the power and magnification of an microscope. So thank you very much for the listening. Thank you very much.